Well, good day to everyone joining us and welcome to today's X-Talks webinar. Today's talk is entitled Advancing MS Clinical Trials, Integrating Medical Imaging, Precision Motion Analysis, and eCOA data for enhanced outcomes. My name is Ryan Muse and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes and this presentation includes a Q&A session with our speakers. Now, the webinar is designed to be interactive and webinars work best when you're involved. So please feel free to submit your questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel, which is on the right-hand side of your screen. And if you require any assistance along the way, you can contact me at any time by sending a message using the same chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, know that all participants are in listen-only mode, and please note that the event will be recorded and made available for streaming to those who registered on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading healthcare research and technology company that generates the highest quality clinical evidence for their pharmaceutical, biotech, and medical device partners. They offer comprehensive evidence-based solutions that combine eCOA, cardiac safety, medical imaging, precision motion, and respiratory endpoints. Clario's science, technology, and operational experts have helped deliver over 26,000 trials and contributed to over 800 regulatory approvals in more than 100 countries. Now I'd like to introduce our speakers for today's event. Dr. Madhura Engelhalliker is an expert in MR neuroimage acquisition, processing, and analysis. Her clinical trial work on imaging endpoints targets the development of novel techniques in diffusion MRI analysis and connectomics, neuroradiomics, and AI-based analysis as well applied to multiple sclerosis, Parkinson's disease, multi-system atrophy, Duchenne's muscular dystrophy, autism, and brain tumors. Laura Khurana is an experienced public health and clinical research professional with a special interest in electronic clinical outcome assessments for clinical trials. Laura has 17 years of experience in the clinical research industry, initially working in web-based data collection for patient registries and post-approval safety studies. And Dr. Adam Jagodinsky is a scientific advisor and oncology lead in the Precision Motion Science and Consulting team, where he works to advance the use of digital health technologies in clinical and academic research. Dr. Jagodinsky consults with sponsors on implementing wearable sensor solutions in clinical trials and provides comprehensive scientific support surrounding instrumented test and endpoint selection. But now, without further ado, I'd like to hand the mic over to our first speaker for today, Dr. Madhura Engelhalliker. You may begin when you're ready.
Thanks, Ryan, and hello, everyone. Thanks for joining with us and welcome to the webinar. Our agenda today includes overview of multiple sclerosis, followed by medical imaging in multiple sclerosis and MS clinical trials. We will then move on to understand the eCOA processes, which comprise of data collection and monitoring, followed by precision motion analysis, and finally we'll wrap it up with the question and answers. Next slide, please. We all know that MS is an autoimmune disorder of the central nervous system, which involves inflammatory damage in the brain and the spinal cord. Myelin sheath normally protecting the axons is damaged, which is shown in this picture here. And MS has a significantly high prevalence in females compared to males, and it is most commonly manifested between the ages of 20 to 40. We really do not know yet the exact reason behind the origin. However, it's likely a combination of genetic and environmental factors. In majority of the patients, MS manifests in relapsing remitting course, which might eventually progress to secondary progressive MS. Diagnostic testing includes MRI of the spine and the brain and CSF testing for oligoclonal bands. Common symptoms are neurological and can include visual, sensory, motor, and cognitive changes. Can we go to the next, please? Now I'm going to talk about the subtypes of MS. MS can be divided into 4 types. The first one is the most common one, which is called as a relapsing remitting MS, where temporary period called relapses or flare-ups are seen. These attacks are followed by periods of remission, where the symptoms may disappear or subside. Remissions can last anywhere from months to years. And approximately 85% of people with MS are initially diagnosed with relapsing remitting type. Um, RRMS may advance to the secondary progressive stage, and this type is characterized by slowly worsening symptoms and neurologic function. The third type is primary progressive MS, and the fourth one is the progressive relapsing MS, which are generally rare and not seen in much of the population. Slide, as we go to the next slide, the treatment is multifaceted and includes lifestyle modifications and general health care, treatment for acute exacerbations, disease modifying therapies, and symptom management. Today, we will focus mainly on the aspects of clinical trials for disease modifying therapies. Uh, as we go on to the next slide, I will start discussing the imaging aspects and imaging trials in MS. So firstly, we will go to the T1 lesions and T1 black holes. So this is a sign which is always seen on the MRI. So T1 weighted MRI measures the longitudinal magnetization and provides excellent structural definition, such as the contrast between fat predominant structures such as myelin that are seen as bright and water predominant structures such as cortex that appears to be dark. In an acute inflammatory phase, when a contrast tracer such as gadolinium is given during the MRI and T1 imaging is performed, a localized CNS inflammation which may occur with the breakdown of the blood-brain barrier and extravasation of gadolinium contrast into the surrounding parenchyma can be seen. So this basically appears as a small enhancing lesion as shown in the first picture here. Presence of enhancement is generally a marker of ongoing disease activity. Active lesions enhance for 2 to 6 weeks and slowly the enhancement goes away. Now T1 black holes, which is shown in the second figure here, these are hypointense lesions and these can be seen on a standard T1 sequence without the tracer. These indicate the chronic stage with white matter destruction, axonal loss, and irreversible clinical outcomes. The active enhancing lesions on the T1 eventually become inactive and can convert to black holes as shown in the figure number 3 here. So you can see the small here enhancement which has gone to become a black hole over time. Going to the next slide, we will look at other types of lesions, which are basically the T2 lesions. So T2 lesions are generally observed on 3 different contrasts. One is the T2 or the flare or the proton density weighted images. Now what they measure the transverse magnetization and hyperintense lesions are generally observed on these images. These lesions reflect chronic inflammation generally and demyelination, axonal loss, and edema. Now images here show new and old both type of lesions, and occurrence of new lesions is always consistent with new areas of um, multiple sclerosis related tissue damage. So here you can see a picture where after 1, 2, and 3 years, the lesion size has increased over time. And the top picture shows the T2 and the flare images both, where hyperintense lesions can be seen in the white matter distinctively. Going to the next slide, we turn our attention to MS clinical trials now. We can see that imaging is a crucial part of the trials. It is needed for eligibility, safety, in particular for monitoring the progressive multifocal leukoencephalopathy, and for efficacy, where we count and measure the size of lesions, quantify brain atrophy over time, and also perform exploratory analysis, which include advanced MRI sequences such as diffusion tensor imaging or magnetization transfer imaging, etc. Going to the next slide. So I go to the point where we are looking at the efficacy outcomes. The first part is the lesion counting. So presence of enhancement is a marker of ongoing disease activity, as we talked about it before. Outcome variables for T1 enhancement include number of contrast enhancing lesions per scan or a cumulative number over time. And correlation between then we going to the T2 lesions. The correlation between the T2 lesions load and clinical impairment are generally poor. However, change in number and the volume of T2 lesions have been used as a surrogate marker in the clinical trials of new therapeutic agents. Also, the location of T2 lesions is correlated with clinical outcomes and can be a crucial marker. Going to the third point, which is the black holes, the relationship between black holes number and volume and clinical features is not completely understood yet, but the change in volume over time is certainly an important marker of pathological progression. Outcome variables include number of new black holes, total number of black holes, and volume of each black hole. Going to the next slide, now how do we compute the volume of these lesions is through a completely automated AI-based lesion segmentation algorithm. So we have trained a deep unit model to segment the lesions on the T2 flare as well as on the T1 to get the black holes volume. The output of the AI model is shown here. In the first picture, you can see a brain with T2 lesions. In the second column, it's a manual segmentation that is performed by a radiologist. And the third column shows the AI-based segmentation, which basically shows here that it is very well validated with the manual segmentation. On the second picture here, you are seeing a T2 image again. Sorry, the first one was flare. This is a T2, where you can see the manual segmentation again and the AI-based segmentation to be almost matching to each other. Similarly, for black holes, you can see manually hand-drawn picture here of the volume of the black holes, and here is the AI-based segmentation. So the accuracy of segmentation or the dye score kind of ranges between 85 to 95%. And we have been using these automated AI-based lesion segmentation tools in our clinical trials. Going to the next slide. Another important aspect in multiple sclerosis is the change analysis. So when a patient comes at the first time point and then it comes for a follow-up visit, the lesion load has changed, and how do you compute that change is through something called as the change analysis, which is performed using automated image processing techniques. So what we do here is make sure that the visits, the images of the visits match each other through a robust registration algorithm. And once they match, one visit is subtracted from the other pixel by pixel so that we can get a difference image. Now, new lesions on the difference image will appear bright, and the resolving lesions are start appearing dark. So this is how we do the change analysis to understand how the change has occurred over time on the T2 images. Going to the next slide. Brain atrophy is another very important component in clinical trials for MS. So here you can see a picture of a patient from 1995 to 2005, and in 10 years there is a huge atrophy. So you can see the ventricles enlarged, the white matter shrunk. So all this can be automatically computed using something called as tensor-based morphometry. And we can get a very good idea of between the visits how much atrophy actually occurred. Now, the only caveat when doing this analysis in MS is that the images do have lesions in it, which can throw away or give erroneous results. So to remove that, we generally do something called as synthetic filling of these lesions. So here it is shown how the synthetic filling can be performed. So if you have lesions here, those are filled synthetically, and then you get a brain which without any lesions. And this image is then used for atrophy calculations. So this basically only makes sure that the atrophy is calculated and you're not using anything of the lesions for your atrophy. So also we compute something called as the normal appearing white matter mass, and then we subtract the T2 lesions mass from this white matter mass basically to get the lesion load. So going to the next slide. Here I'm talking about the advanced MRI sequences that are used in MS clinical trials. So the first one is the diffusion MRI, which provides a superior contrast in the white matter and also is very important for quantifying the molecular, the microstructure of the gray matter. So here the analysis includes estimation of diffusion tensors or any higher order models such as bitensor or neurite orientation, dispersion and density imaging, etc. And once we calculate like the scalars such as fractional anisotropy, mean, axial and radial diffusivity in the regions of interest, such as T2 lesions, NAWM or other ROIs. This can help understand how the white matter structure and integrity is changing over time. Similarly, we can also use magnetization transfer imaging, which is basically a T1 contrast, but used with the magnetization transfer on and off. Then these two images with the on and off are registered or matched with each other. And then a magnetization transfer ratio map is calculated. Now, this ratio is important to quantify the macromolecular density and therefore the tissue damage. So this is what is shown here. Finally, going to the spine part of MS, we have, going to the next slide, we have the spinal cord volume analysis that we perform. So this is an automated cord segmentation using the spinal cord toolbox on T1 or T2 images. And this is performed using the spinal cord toolbox, and it automatically labels the vertebrae. If the vertebral labeling is incorrect, you can go in manually and define the upper cutoff, that is the inferior border of the cervical one, and then basically it can again calculate the vertebral labeling. And this also helps in performing calculations for the cord volume and cross-sectional area in millimeters square, and gray white matter segmentation using the dedicated axial scans of the spine. So this is what like imaging of MS in clinical trials includes. And with this, I would like to end my talk and give it to Laura Kurana for the eCOA data collection and monitoring.
Thank you, Dr. Inga Holliker. From imaging, we're going to change gears a bit and talk about eCOA and what role this type of data plays in drug development programs and clinical trials in multiple sclerosis. So first, I want to start with defining some of the acronyms I'll be using. COA stands for Clinical Outcome Assessment, and the FDA defines this type of data as a measure that describes or reflects how a patient feels, functions, or survives. And when those measures are collected electronically without a paper source, we call that eCOA. COAs include a few different categories: patient-reported outcomes, or PROs, clinician-reported outcomes, or ClinROs, performance outcomes, PERF-Os, and observer-reported outcomes, OBSROs. COAs are inherently more subjective than the other types of data we're discussing in this webinar, and they collect data that can be much more meaningful to patients, and they can be an important part of the endpoint strategy for a trial of MS treatment. We often see them supporting efficacy endpoints, especially disability, function, and quality of life, and they can also support safety endpoints like suicidal ideation and behavior. Go to the next slide, please. So if you plan to collect eCOA data in your MS trial, you'll need to think about when, where, and how to collect that data. And on this slide, we're showing in the two horizontal rows that this data can be collected at visits to the clinical trial site at the bottom of the slide or between site visits. And when you think about how that data should be collected, often the choice is between paper and electronic. And the goal here obviously should be to maximize site and subject compliance with completing these assessments so that you can maximize your data quality. We know that regulators, including the FDA, expect trial sponsors to collect COA data following what's called the eCOA principle. And that acronym stands for attributable, legible, contemporaneous, original, and accurate. This was a framework that was developed in the 90s to ensure the accuracy, reliability, and completeness of data. And it has since been expanded to include several other factors. But the key thing to remember here is that it's much easier and you're much more likely to meet these principles when you collect your data electronically versus on paper. Next slide. And while you're thinking through your strategy of when, where, and how to collect your eCOA data, you should also keep in mind the relevant regulatory guidances and best practices that have been published in the field. And I've included a few of those here on this slide. The first one on the left is from the EMA. This is a scientific guideline on clinical investigation of medicinal products for the treatment of MS. It was published in 2015, and it talks about the endpoints that are appropriate and acceptable for MS trials. It specifically calls out endpoints around relapse, including annual relapse rate, and endpoints based on prevention of disability progression or delay of disability progression. It specifically calls out the EDSS, which is the most widely used and well-known scale to assess a patient's level of disability due to MS. When it's used in trials across time, the change in disability due to MS can be measured. Another important professional and scientific body is the Multiple Sclerosis Outcome Assessments Consortium. This was launched by CPATH back in 2012, and it's a partnership between MS advocacy groups, regulatory bodies, government research groups, academia, and industry. And they've done some great work creating a database of patient records from placebo arms of 9 different MS trials, and this data is available to qualified researchers. They have also done systemic reviews of the literature to summarize the evidence for the validity of some of the most commonly used eCOA assessments in MS. Next slide. On this slide here, I've outlined many of the common COAs that are collected in MS trials. On the left are the ClinRos, PERFOs, and composite scores that we often see supporting endpoints in MS trials. The most important one in this category is the EDSS. I've mentioned it already. I'll talk a little bit more about it in my next slide, but another ClinRo that's often used to support safety endpoints in MS trials is the Columbia Suicide Severity Rating Scale, the CSSRS. For performance outcomes, we have the timed 25-foot walk test and the 9-hole peg test, which assesses a patient's level of hand dexterity in placing pegs into holes on a tabletop device. There are also several composite scores that have recently gained traction as endpoints. The EDSS Plus involves combining the EDSS score, the timed 25-foot walk test, and the 9-hole peg test. And the MS Functional Composite involves combining the paced auditory serial addition test with the timed 25-foot walk test and the 9-hole peg test. And on the right side of this slide are the most frequently used patient-reported outcome measures, including a patient-reported version of the CSSRS. So these COAs serve an important role in MS trials because they provide data on disability, symptom burden, cognitive ability, quality of life. These are all measures that are often very meaningful to patients, and trials can track this data over time to show how these measures change in response to treatment to determine treatment effect. Next slide. So I have mentioned the EDSS a few times, and I wanted to spend a few minutes going into more detail about this assessment specifically because it does play such an important role in MS drug development programs. EDSS stands for Expanded Disability Status Scale. It was first published in the 1980s, and it has since gone through several revisions and improvements, but it remains the gold standard for measuring a patient's level of disability due to MS. And if there's one thing I want you to take away from this slide, it's that there are 2 versions of the EDSS. The first one I'm showing here is the Kurtzky EDSS, and it's comprised of 9 different data points. So 8 different functional system scores, and then the EDSS step. So the clinician performs a neurological exam on the patient. They come up with a summary score for each one of these functional systems: visual, brainstem, pyramidal, etc. And then the EDSS step is a single overall summary score of that patient's current level of disability due to MS. So because it's only 9 data points from an eCOA perspective, it's a relatively simple instrument to build. But this also means that with so few data points summarizing such complex body systems, this version of the EDSS has low inter-rater reliability, and there can be quite a bit of variation in how this tool is actually applied from rater to rater and from patient to patient. The Kurtzky EDSS is in the public domain. There's no licensing required to use it in your trial. And we frequently see this version of the EDSS used in phase 1 studies or when it's supporting an exploratory endpoint. So Ryan, if you click one more time, then we'll see the other version of the EDSS called the Neurostatus version. This one was first developed in 2004, and it was intended to address some of those scientific concerns around the inter-rater reliability of the Kurtzky version. So it asks the clinician to document more data points in each of the functional systems during that neurological exam. And that amounts to about 120 different subscores that the rater enters, which are then summarized by the overall functional system scores, and then finally summarized by the single EDSS step. The Neurostatus version is much more complex to implement in an eCOA system, not just because of the large number of data points, but because there are complex scoring algorithms that are applied to these data points to improve data quality. This assessment is under copyright by Neurostatus University Hospital Basel, and they have a required expert review process whereby their neurologists will review any forms that are submitted by study sites, any EDSS forms that contain inconsistencies identified by that scoring algorithm. So all of these features have substantially increased the inter-rater reliability of the EDSS, and the Neurostatus version is more scientifically robust, and it's the gold standard for phase 3 studies and any endpoints that are supporting regulatory claims for efficacy. So if you're running an MS trial, it's very likely that the EDSS will be included in your protocol. So it's helpful to know that there are 2 versions of this assessment, and you'll need to do a cost-benefit analysis and consult with eCOA experts who can help you decide which one is best for your trial. Next slide. So MS trials face several unique challenges in collecting COA data. Firstly, the endpoints themselves tend to be complex and subject to rater variability, which we've talked about already. These complex assessments that have been around a long time can present a challenge when migrating them from paper to electronic format. So you want to make sure that you're going with an eCOA vendor with strong solution design practices and experience in designing and implementing these assessments. And because of the nature of many of these ClinROs where raters are asked to assess and score MS patients, they're inherently vulnerable to rater variability, where the investigators in your trial don't have concord with each other on how they're administering the scale. So rater qualification and training is especially important. All these assessments are intended to be administered in your trial. We also encounter sometimes some resistance at the site level to eCOA systems, again, with investigators being familiar with the paper versions and reluctant to try something new. So having a well thought out eCOA solution design can help with that, and also keeping in mind that ultimately direct electronic collection is going to increase your data quality and integrity. And finally, MS trials often have complex inclusion-exclusion criteria that can be difficult to operationalize at the level. This can sometimes lead to delays in patient enrollment and in getting trial subjects identified and qualified. So anything that can be done to improve that workflow at the site level can help with patient enrollment. So I'm thinking about things like eligibility reports that automatically calculate complex scores for the investigators and give them real-time feedback about eligibility. Next slide. There are some general takeaways that you can keep in mind when looking to implement eCOA in your MS trial. You know that ClinRO data, clinician reported outcome data, will be playing a key role, and those assessments are often complex. So make sure that you're working with partners who have experience in implementing them electronically. And finally, training is crucial, both for raters and to ensure that they're concordant with each other, and also for participants to ensure that they know what is expected of them and can plan for successful participation in the trial. That was a quick overview of clinical outcome assessments in MS trials, and I'm now going to hand it over to my colleague, Dr. Adam Jagodinsky.
Thanks, Laura. All right, next slide, please. All right, so here we're going to talk about just some different types of clinical evidence for determining the best types of digital gait and balance endpoints for MS trials. And first, one key area is looking at whether these digital measures for gait and balance are sensitive and specific to MS. And so we'll show some studies that identify specific gait and balance measures that can best distinguish MS patients from healthy controls, and also can distinguish between MS subgroups. And it's also important to assess how gait and balance endpoints relate to a neurologist's assessment of disease severity, and if measures are related to patient-reported outcomes, as this is a reflection of what's most meaningful to patients. And so finally, best gait and balance endpoints should show responsiveness to intervention and reflect progression of neurodegeneration. And we'll discuss some of these aspects as well with respect to various imaging parameters in the later slides. Next slide, please. All right, so this slide shows Clario's Opal wearable sensor solution to capture digital gait and balance endpoints for clinical trials. And this system can be configured for up to 6 sensors. And these are synchronized sensors that capture precise digital endpoints of movement during prescribed tasks. So you'll see, for example, several common gait tests that are implemented are the 2-minute walk test and the 6-minute walk test. We can also capture turns with the gait assessment. The balance is a simple 30-second sway test that can involve several conditions, as you see, with eyes open or closed, feet together apart to challenge the sensory systems. And the third is mobility tests, such as the tug, sit to stand. Those are the 2 of the more common tests for mobility. And these tests can also be paired with cognitive assessments and dual task paradigms as needed for particular subsets. Next slide, please. So when we talk about gait and balance endpoints, we're not just referring to gait speed, but also measures that span across several different domains of gait. And this includes a temporal or rhythm domain. For example, the duration of the step is a measure in the temporal domain. The variability domain measures stride-to-stride variations, and those are clinically relevant changes and variations that we can assess with instrumented gait. Asymmetry can be assessed by comparing movements of the left and right extremities. And stability refers to sort of the trunk motion, upper body motion, and sway as well. And then last, turning is an important domain that can be assessed during gait and mobility tests. And so the pink boxes here indicate the gait domains that are most sensitive for MS, and we'll cover some of the supporting evidence in the next slides here. Next slide. All right, so one of the most common gait tests in MS studies is the 6-minute walk test, which is used to assess the physical function and performance and fatigue as well. And this type of test usually uses a tape measure with floor markings and a stopwatch to measure a distance. But this is an example of a study that conducted the 6-minute walk test with subjects instrumented with the Opal sensors. And here, distance walked was derived from the sensors, and the results showed a group difference comparing 25 MS subjects to healthy controls. So in this case, the study looked at distance walked here, but there's numerous endpoints that can be captured from the sensors. In this particular case, 6 sensor configuration was used with 2 sensors on the feet, 2 on the wrists, 1 on the chest, and 1 on the lumbar or lower back region. And so in the next slide, we'll kind of look at some additional measures that are relevant here. And this study had patients complete a 6-minute walk test with 3 Opal sensors configured to the lower extremity and lower back. And they found that people with a severe and moderate MS had less rhythmic gait compared to controls, and found that people with severe MS had less rhythmic gait and more variable gait compared to people with moderate MS. And so one additional finding that relates back to kind of what Laura was discussing in the eCOA realm, the gait measures in the rhythm and variability domains were shown to be moderately correlated with the EDSS scores, patient-reported outcomes. So in another study, if we move to the next slide, 6-sensor Opal configuration was implemented for gait analysis and compared to the stopwatch timed 25-foot walk test. And the figure on the left shows no difference when comparing walk time between MS patients and healthy controls. However, the instrumented gait analysis was able to show differences in both trunk range of motion, which is shown on the top bar graph, and then turning duration as well, which is shown on the bottom bar graph. And so in another study on the right-hand side, so from a separate study, results showed that prediction of the self-reported walking limitations, so MS-WS12, over 12 months was significantly improved with the 6-minute walk test turning parameters added into modeling. So really this slide is highlighting the sensitivity of Opal-derived trunk stability and turning measures in MS studies above and beyond typical timed walk test outcomes. In the next slide. So in addition to the gait measures, this slide shows sensitivity of balance measures obtained using an Opal sensor on the lower back during a 30-second standing balance test, in this case with eyes closed. And so that's kind of shown on the top left. You can see the figure is standing with feet kind of in a natural stance position with the sensor placed on the lower back. In this case, the eyes were closed to remove the visual input and tax the motor system. And so in this case, the RMS sway measure, which is shown in the pink box here, showed sensitivity, not just distinguishing MS patients from controls, but also moderate versus mild MS patients in a dual task paradigm. So this highlights the utility of sway measures here as well. And the next slide will also show some results from balance tests. And so when comparing MS patients with healthy controls, in this case, we're looking at sway dispersion. So again, kind of pointing to the figure here, the sway dispersion can be kind of visualized on the bottom left in a control group, much tighter sway pattern compared to the MS group, which has much larger deviations anterior, posteriorly, forward and backward, and then side to side. And in this case, the bar graph is showing that sensitivity of the sway dispersion in the left to right direction, comparing MS versus controls. And again, this was sensitive when the clinician timed 25-foot walk test did not show differences between group. Okay, next slide, please. And this slide shows, looks at the association between balance impairments and somatosensory pathway integrity as measured by the radial diffusivity using DTI technique, as Madhura touched on earlier. So as myelin degrades, this disrupts the signaling from the periphery to the central nervous system, and that is an important aspect for maintaining balance. So this plot shows a correlation between balance impairment and reduced somatosensory tract integrity in people with MS. And in the next slide, we'll also look at spinal cord cross-sectional area and how that relates to some of the sway measures here. So on the left-hand side, this is just looking at comparing two different conditions of sway tests that can be implemented. And you can see the with eyes open, EO, compared to eyes closed conditions. The eyes closed condition was created a much larger postural sway response in the MS compared to the eyes open condition. However, when looking at how these measures correspond to measures of spinal cord area, the eyes open condition produced a strong, showed a strong correlation with spinal cord cross-sectional area. So two measures, the sway jerk and RMS sway showed significant correlation to spinal cord cross-sectional area. And then on the bottom chart, on the bottom right, we see the eyes open sway measures, RMS and range were significantly correlated with the change in spinal cord cross-sectional area over a 2-year period. So we see that the sway measures not only are sensitive, but also correspond back to some of the imaging endpoints that we discussed, that were discussed previously. Next slide, please. Okay, so this study, this last study demonstrates the utility of daily life, gait and activity monitoring, and these measures that are obtained from the home environment. So it's important to note that the daily living gait does not involve prescribed walking or balance testing, but rather this is capturing the functional capacity in the home. And this is inherently different from in-clinic performance testing and considered a sort of a complementary approach to in-clinic testing, but not necessarily a replacement, as these two data sets, types of data, are not comparable. And this study showed both activity and lower body gait domains captured during daily life show sensitivity to MS. And the top measures included in this analysis were included median strides per walking bout, percent time in double support during gait, stride and step duration as well. And then looking at the scatter plots, this is highlighting the significant correlation between the double support measure and with EDSS scores. So there's a link there to sort of patient meaningfulness and these measures that are captured from the home environment. And next slide. So in a summary here, the gait and balance measures are highly sensitive to MS and they're meaningful to patients, showing correlation with patient-reported outcomes. Turning speed is not commonly assessed clinically for MS trials, but it is more sensitive than gait speed. And postural sway measures are also very sensitive and reflect the process of neurodegeneration in MS. And last, the daily life monitoring of gait and activity can provide some additional insight into a patient's functioning at their home environment without the presence of an administrator. And these measures are also sensitive to MS and complement the in-clinic assessments as well. All right, next slide, please. And wrapping up here, so we presented on several avenues for integrating the imaging, gait and balance, as well as ePro data in clinical trials for multiple sclerosis. And this includes potential to integrate AI advancements for monitoring lesions and imaging approaches, as well as incorporating well-designed eCOA and digital gait and balance assessment strategies for MS trials. And with that, thank you all for joining, and I will pass back to Ryan now for the Q&A portion of the webinar.
Well, thank you very much, all of you, for this very insightful presentation. I'd like to invite our audience to continue to send their questions or comments for the Q&A portion of the webinar. If you're attending via GoToWebinar, you can use the questions window. For those of you watching on LinkedIn Live events, feel free to post your questions in the comments section. I've already received some questions from the audience though, so we'll get ourselves started with those. The very first question I have for you would like to know, do I need to standardize image acquisition across sites, or are local standard of care protocols good enough?
Hi, I can answer this. So yes, we definitely would. For clinical trials, we expect that we standardize the image acquisition across sites so that we are consistent across sites, and the quantification that we are doing for lesion segmentation, for lesion counting, and for atrophy measurements are can be compared across sites as well. So we definitely would want to make sure that there is a consistent image acquisition protocol that is used. Now, for how do we do this is, so for example, there are say 10 sites in the clinical trial, then we generally send them out the protocol, which they can import it on the scanner and they can use it directly on the scanner and scan the patients using that protocol. If we don't have the protocol for, say, the scanner is really new and we don't have the protocol for that scanner, in that case, we also send them a quick reference guide where we have listed all the MRI parameters such as the TR, TE, what type of sequence, what is the RF pulse, what is the flip angle, and so on. And everything is being set. Once it's set, then only we approve the protocol, and then that can be used for the patients.
Wonderful. All right. Thank you very much for that answer. The next question I have here would like to know, well, it says that first, I didn't know there are two versions of the EDSS. So what they're curious is, how can I decide which one is best for my trial?
I can speak to that. So as I mentioned before in the eCOA section, the EDSS is a single score from 0 to 10 with half unit increments that is used to summarize a complex concept, the overall level of disability that a patient is experiencing due to MS. And the two versions of the EDSS represent two different approaches that you can ask your raters to take to arrive at that single score. One's more complex and detailed than the other, but it's more scientifically rigorous and allows for more standardization in administration of the assessment. So as a general rule, in phase 1 studies, when it's supporting an exploratory endpoint, it's appropriate to use the simpler Kurtzke version of the EDSS. In phase 3 studies, it's advisable to use the neurostatus version. Phase 2 is where it gets a bit tricky to make that call, but this is something that you should discuss with your eCOA vendors' scientific team so that you can arrive at a decision that makes sense for your trial.
Perfect. Thank you very much. The next question I have is wondering about clinical tests of postural sway or balance are not commonly implemented in MS. So what is the value of adding a sway test for clinical trials in MS?
Thanks, Ryan. I can jump in. So the balance and sway are regulated by pathways that travel through the spinal cord into the somatosensory cortex. And when these pathways deteriorate in MS, that leads to impaired balance and more postural sway. And so these types of balance impairments are meaningful to patients. They affect quality of life and they relate to fall risk as well. And so the instrumented sway tests are really relatively quick, around 30 seconds, feasible for large scale trials. And there's a wealth of endpoints that can be captured in this simple test, and with a single sensor on the back. So these are accurate, reliable, and sensitive to MS and show good potential for MS trials.
Perfect. Thank you very much for that answer there. The next question here is wondering about what are some common brain regions where lesions related to multiple sclerosis are observed?
Hi, I can answer that. So as I showed in the presentation, we can see lesions on different types of lesions. We see on the 3D T1 contrast, which are the enhancing lesions, but we also see the T2 and flare lesions and black holes. Now, most of the T2 and flare lesions which are seen are around the small veins in the brain. These are generally located in the periventricular areas, like I showed in the image before, like from year 1 to year 3, the the lesion size actually increased in the periventricular region. These can also be located on in cortical regions, infratentorial regions, and the spinal cord. Some common areas are also seen mainly on the optic nerve and especially in some brainstem regions as well. So yeah, I mean, they can be everywhere, basically, but they're seen mainly around the small ones in the brain.
Wonderful. All right, thank you. Next up here, someone's wondering what kind of accommodations are possible for making eCOA accessible to everyone. Specifically, they're thinking about MS patients who may have difficulty using a tablet or a smartphone due to visual problems, for example.
Yeah, that's a question we get a lot, not just for MS trials and MS patients, but in other indications and therapeutic areas as well. For patients with vision problems, we can adjust the font size on the eCOA device on the screen. We can do that across the board so that all assessments for all patients in the trial see a larger font size, and it makes it a bit easier. We can also implement functionality like the pinch and zoom functionality you may be familiar with on your iPhone or any other touchscreen device, where patients can zoom in and zoom out on their own at their own prompting. And patients can adjust that themselves. Our eCOA app, Clario's eCOA app, can also accommodate dark mode, which is you may be familiar with on your iPhone as well. And all of those things can help with visual accessibility. In some cases, I'm thinking about other accessibility accommodations too. It might be appropriate to use an interview version of a patient-reported outcome tool when those are available. There are lots of different accessibility options, and when sponsors come to us with those questions, we can present those options. We can choose together what makes the most sense for the patient population that's in this trial.
Fantastic. All right, thank you very much for that answer and for all of the answers today, but we have reached the end of the Q&A portion for our webinar. For those interested in hearing more from Clario, you can register for their next webinar, Psychiatric Drug Development, Digital Endpoint Solutions to Extract the Most Value from Your Clinical Study, which is scheduled for Wednesday, October 16th, and you can use the link I've sent in the chat box. Also, if we couldn't attend to your questions, know that the team at Clario will follow up with you, or if you have further questions, you can direct them to the email address that's up on your screen. I want to thank everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen as you exit, and your participation is appreciated as it helps us to improve our webinars. Additionally, I've shared a link to view the recording of today's event in the chat box, which you can share with your colleagues so they may register for the recording here as well. And for those watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join me in thanking our speakers for their wonderful time here today. We hope that you found the webinar informative. Have a great day, everyone, and thank you for coming.
Thanks all.